please. So welcome. It's Mitya's second lecture. So please, Mitya.
Hello, everyone, and welcome to the second lecture on the topic of normative approach to neural computation, where our goal is to derive a mathematical algorithmic framework that would describe what neurons do on the level that would be appropriate with the recently available large-scale connectomics and neuronal population activity imaging data. So a quick recap of what we did in the first lecture. We started with this normative approach where we have to guess the objective function. And the objective function that we used was taken from what has been done in neuroscience for 40 years, which is a reconstruction or decoding approach. And that approach allowed us to derive some pretty simple but insightful results into neural operation that could also be confirmed experimentally. But when we tried to generalize it to multi-component principal component analysis, we ran into difficulties in that the learning rules were not local. And here is the slide that we finished with. And just to make sure we're all on the same page, the problem was that when we derived a learning rule to update the feed-forward synaptic weights in the circuit below, what happened was that there was this summation over k, which required the activity yk of all the other neurons in this layer.
Mitya, you're still on your first slide. Is that normal?
I'm sorry?
No, he's on his second slide.
Okay, now it's switched to the second one. Okay, sorry. I was.
So this summation requires for the synapse that connects just two neurons know the activity of all the other neurons in the system. And that's, of course, biologically implausible. And that's why we said that this approach ran into problems. We would like to circumvent this by guessing a different objective function, and that's what we will do today. But before I do that, I just want to point out one other issue with the reconstruction approach that has a little bit of a philosophical nature, but is already problematic. And this has to do with the invariance of concepts. So, we all communicate with each other, right? And such language communication requires the invariance of the meaning of the words that we use, right? So it's not clear how such invariance could be maintained by neural correlates in the brain. For example, if I say the word red, how do we know that everyone thinks of the same color, right, when I say the word red? And this reconstruction approach doesn't really help to answer this question, because in the reconstruction approach, if I have a stimulus red, stimulus X being red, there is some kind of activity pattern Y that reconstructs that stimulus. But that Y cannot be the same in each of our brains. This is because neurons in mammalian brains are not identifiable. The circuits are exactly the same maybe in the fly visual system, but certainly not in mammals. Even the number of neurons is different from individual to individual, and you can see differences even on a large scale, like using MRI recordings. So it can't be that the same neurons respond to the word red in every brain. But if these are not the same neurons, that what is it there to guarantee that the meaning of the word red are represented in each individual's brain is the same. So this kind of question also, I want to bring it up because it kind of it is problematic in the reconstruction approach. Okay. Anyway, but because of these problems with the reconstruction approach, we decided to
Nitya,
yeah.
could I just ask at this point, that's a very, very interesting. I want to ask about another phenomenon in language, and I want to ask whether the problem is of the same caliber as what you just said just now. So other than, yes, there should be invariance, but often in not often, like in language, for example, phonological categories. So we have to distinguish the sound categories, right? The sound p and the sound b. And what we know is that the sound stimuli itself is never always identical. So what our mind is actually always taking it is always like a category. So it's not exactly of the same stimuli, but in fact, we have to always categorize over a certain range of a sound. So what I want to ask you is that, does that make the invariant problem in terms of the neurons from the reconstruction approach, like less good or less bad?
Yeah, I mean, this problem of, nuisance variables, right? That, some sort of changes in the stimulus that don't affect the classification
Right.
result is a very important one. And I actually have a few slides that I can show you what I have to say about that. So it is an important problem. It's not immediately clear to me whether reconstruction approach has a particular disadvantage in addressing this. But I do use it as a way to show off the advantage of the similarity approach. So maybe, yeah, you're probably right. I'll come back to that
Great, thanks.
probably in the second half of the lecture. I will use a visual stimulus example, but it's the same idea. So maybe we can, you're probably right.
Right, thanks.
Any other questions? Okay. So the approach, so basically, we have to go back to the objective function and guess a new one, and then repeat the process. In the objective function that we use, we call the similarity matching approach. And before I explain exactly what it is, let me show you some experimental data that will suggest that thinking in terms of similarities is actually advantageous and may correspond to something that happens in the brain. So this data come from imaging in the fMRI scanner, where a subject in the scanner is shown 100 different images. And, you start with faces and body parts and household objects, at least if you live in the United States, and all kinds of images. And each image elicits a vector of activities. But unlike before, each component of that vector is not an activity of individual neuron, of course, because of the coarse spatial resolution of fMRI. It's an activity of a voxel which contains 10 to the 5 neurons. Nevertheless, you can come up with an activity vector, and then you can compute inner product or correlation or scalar product between all the pairs of vectors, activity vectors, elicited by different objects. And this is what is shown in this diagram by this color scheme, where dark blue color corresponds to the highest similarity of the activity vectors. And of course, that happens in the diagonal because the vector is most similar to itself. But in addition to the diagonal, you also have this block diagonal structure, this blue squares, that tells you that the activity vectors elicited by two animate stimuli are more similar to each other than the activity vectors elicited by animate and inanimate, the red squares. Okay? So this similarity plot tells you that the brain, at least in higher cognitive areas like IT, cares about the categorical similarity of visual objects, such as animate and inanimate. Moreover, if you repeated this experiment in different individuals, you will get a very similar, although not identical, similarity matrix. So this similarity matrix is preserved among individuals. And there is even more. When they computed the same similarity matrix in a macaque monkey, in the IT area of macaque monkey brain, and this one they obtained using electrophysiology, using single unit recordings. Okay, so now the activity vectors are indeed neuronal activity vectors. But when they computed the similarities between all pairs of image elicited activity vectors, they got a very similar similarity matrix to what was obtained with fMRI in a human. Okay, so not only similarity matrix seems to be rather invariant among individuals, but it is somewhat preserved even across species, which is already a very interesting result. But in our context, it proposes a possible answer to this philosophical paradox that I outlined in the beginning of how we maintain invariance of concepts. Okay, so if you take this similarity data into account, it's not that the word red elicits the activity in the same pattern of neurons in each individual's brain. It may be a very different pattern of activity, but what matters is that that pattern of activity has the same similarity to other related concepts to red, such as, colors orange and pink, and maybe the word blood. So these kind of similarities between activity patterns in the brain are preserved across individuals. And that is what's responsible for the invariance of concepts between all of our brains. Okay? So the importance of similarity as opposed to individual patterns in neuroscience has been recognized before. For example, it led Shimon Edelman to claim that neural representation is representation of similarities in the sense that I just mentioned. So we took this a little bit further and explored the hypothesis that neural computation is the transformation of similarities. And what's the simplest transformation? The simplest transformation is just preserving the similarity. So that's what we will start with. We call this similarity matching, and this is the principle that similarity of inputs to a single layer of neural network is preserved in the activity of that neural network. Okay? So that's the principle that we'll use to substitute the reconstruction approach. To make it more precise,
just excuse me, just a conceptual point. Here, similarity depends on what metric you choose, right? And for example, similarities between faces, if you want to describe real similarity between faces, it will be a very complicated metric.
Yes, so this is a very, very good point, and we will get to it in detail. At this point, let's not worry about it, and we will just consider similarity in the simplest possible way. So if we have an image made out of intensity of pixels, then the vector of pixel intensity of image 1, x1, is multiplied by the vector of pixel intensities of image x2 as an inner product or a scalar product, which is just the sum of component-wide products, and that will be our metric of similarity. Okay? I realize that this metric doesn't help you very much to classify or identify who the face belongs to, but we'll get to that in a minute. Okay, so let's just start with the very simple-minded similarity on the level of pixels.
Okay.
Okay.
Thanks.
Any other questions? Okay, so similarity of inputs is shown on the left, similarity of outputs on the right, and I think it's important to emphasize that to avoid the trivial solution where y is just equal x, we have to introduce some kind of constraint. In this case, we'll just assume that the dimensionality of the output is smaller than the dimensionality of the input. So, for example, here I'm drawing a 3-dimensional input and a 2-dimensional output. And the different colored vectors are different activity patterns. And it's important to realize that in the similarity matching approach, we do not require that the identity of the vector remains the same. It can't even be defined because there's a different dimensions in input and output. But it's the similarity which could be approximated by the angles between the red and the blue vector that is the same between the input and the output, or between the blue and the green vectors. Okay, so this is what is being preserved here. And mathematically, we express this through the inner product. And of course, you don't just want to preserve an inner product between one pair of stimuli. You want to preserve all of them. So this is the matrix of all possible t-squared similarities, where t is the number of stimuli presented that will eventually be in the online setting t time steps. But in the moment we're still in the offline setting, so it's just a data set of t stimuli. And then there are t-squared similarities for the input and t-squared similarities for the output. And we are going to set them to be equal. I should also emphasize that the similarities here form a matrix which is called a Gramian or a kernel matrix in machine learning, which is different from the covariance matrix. In the covariance matrix, the summation is over index t, and you have like x I t x j t, and that gives you a covariance element Cij, right? So that's different from what's being done here because the summation is done over the I index, okay? And then for different pairs of t's, then t and t prime, you compute all those similarity pairs, okay? So just like we did in the reconstruction approach, if we want to have two things equal, we can write down an objective function which is minimized when that equality is satisfied, which is simply the difference between the components of these two matrices squared summed over the whole all possible pairs. So this is our new objective function that will replace the reconstruction approach objective. And in fact, this objective function is not completely new. It has been used in a statistical method called multidimensional scaling. And because of that, people actually figured out what the offline solution is for that objective function. Amazingly, that objective function also performs principal component analysis on the data with a little caveat. And the caveat is that the y's that come out of it may be principal components, or maybe some kind of a rotation of those principal components caused by an orthogonal transformation. Okay, so what they say is that this solving this objective produces the top principal subspace of the data covariance, which is includes all the possible rotations of the basis. But other than that, this is the same old PCA that you know and that we use to analyze the reconstruction approach. So despite the fact that we use the different objective function, the optimal solution is actually the same as before. And this is good news because we are going to inherit these successes in explaining experimental data, such as the synaptic weight vector corresponding to the top principal component, which is the eigenvector of the covariance matrix in the Flayl factory system, and the decay of the learning rate with time and cumulative activity that we saw in LTP experiments. Okay, but what we hope for is that, I should also say that in a single channel case, if the dimension of y is 1, it's just a scalar, then not just the solution, but the objective function is exactly the same. Sorry, I shouldn't say that. The solution of this problem is exactly the same as PCA, because you cannot rotate this one dimension, okay? So it is the top principal component. But in the multi-component case, we hope that we will be able to derive the learning rules that are local and therefore biological plausible. So that's the plan. Any questions?
I think we're good.
Okay. All right. So now we have to do the hard part, right? And to derive in the online setting where x's are presented sequentially, right, one at a time, we have to derive the optimization algorithm and then map it to neural network. And why is that a hard part? Well, just by looking at an objective function, some might think that we're actually crazy to use it for the online approach. Why? Because look at these terms. They combine input data from time points t and tau, where t and tau could be at any time, right? So t could be an image you saw when you were 1 month old, and tau could be an image you saw when you were 20 years old. So how is it possible for the brain to optimize this objective? Is it going to keep around the image from 1 month old until you're 20? No way this is going to happen, right? There is not enough memory to store this in the brain. So that can't possibly happen. And also you have to compute the output y online on the fly. So what's going on? How are we going to derive this online algorithm? And that was one of our major contributions, where we pointed out that a simple mathematical, even algebraic trick will allow us to do that. And that trick is based on the observation that when you expand the square, okay, so one of the terms that you get would be minus 2 xt. So I intermittently use the transpose notation and the dot notation for the inner product. I hope that doesn't bother anyone. Okay, so you have a term like this, right? The cross term when you expand this. And this term can be massaged so that the variables that depend on the index tau are within this parenthesis. And therefore the sum can be taken inside the parenthesis as well. And what is that sum? That sum is a covariance between x and y variables, right? So now it is the summation over tau, right? Just as we just discussed. So it's not similarity, it is covariance. And what's a good thing about covariance? Well, a good thing about covariance, if it's a long sum, so you already looked at a significant chunk of the data, then by showing one more data point, is not going to change the covariance that much. And just like we did in the reconstruction approach before, we're going to use this fact to substitute the sum through t by the sum through t minus 1 and use the w, the notation for the sum, from the previous time step. And then keep it constant and minimize with respect to y.
Mitya,
Yep.
another, maybe I'm wrong, but another solution could have been maybe to just introduce a discount factor. And if your discount is an algebraic factor, then you would have kind of an exponential decay of your memory. And you would get to a steady state where you just have to, then I think you could do it online, no?
Yeah, but that's correct, and this is one of the things that you can do if you want to have adaptation and follow a non-stationary distribution, but you
Right.
still have to handle this issue within the time scale of forgetting. And I'm disallowing to store all this data in memory even over that time period, because this could be, thousands or 100,000s of images. So where are they going to be stored?
Okay, but you would store just this sum like you're doing here, right?
Oh yeah. Oh, if you're just saying that I should do this with the discounting factor, yeah. But with the same trick, sure.
Okay, I will have to think more about it because of how the two things converge. Okay,
Well,
thank you.
we can discuss offline, but for this problem so far, that's the main trick, okay?
Okay.
And the same trick can be applied to the quartic term, which is okay. Sorry, okay. To the quartic term, which is yt transpose y tau transpose yt. And then we can take the taus into the parentheses and sum over tau. And then we'll have a similar quantity wyy, which is a covariance of y variables that we can inherit from the previous time step and go ahead with optimization with respect to y. But now this optimization is done on a linear quadratic cost function where this term is linear in y, this term is quadratic in y, and so it's just a convex function. We can easily optimize it by descending along gradient descent using this kind of iteration, which is simply the gradient step. And this iteration is naturally realized by a neural network, which looks just like the one we had in the reconstruction approach, where this matrix vector product is implemented by the feed-forward projections with the elements of the matrix WYX represented by the synaptic weights in those feed-forward connections. And the second term is also matrix vector multiplication, which in the neural network language corresponds to these lateral connections that connect y to y. And they come with a minus sign. Okay? So this circuit requires, of course, iteration. That means that there is once we present a stimulus, activity propagates forward, and then there is some recurrent activity in this network that has to settle, and after that we computed the y's that can be output. But that's just like in the reconstruction approach, only the first phase of the algorithm. There is also the second phase where we adjust the synaptic weights, w's. Because remember, the w we used for the iteration is the one which is taken from the previous time step. And now we have to recompute the w so that it can be used in the next step. And the w here is given simply by this summation, normalized summation of the previous inputs, because this is just like normalized covariance. And normalization comes from the fact that for the diagonal y's, to get the y out here, we have to divide the right-hand side by the cumulative activity, which is just like what we had in the Euler's rule. But the numerator is different. Okay, so it's simply the summation of the previous stimuli, and although it was true in the single component PCA case, it was not the case in the multi-component, and that was the cause of our troubles. Okay, and now, okay, the learning rules, which are just recurrent relations derived from this summation, are shown here, and the beauty of this approach is that these learning rules are local. To make this explicit, I kept the subscripts I and J, meaning that this is a synapse that connects neurons from J to I, and it depends only on the weight of that synapse, the activity of the two neurons that synapse connects, again the postsynaptic activity and the weight of that synapse, and then there is this again this cumulative activity of a postsynaptic neuron, which can be stored in some sort of calcium modulated gene expression variable. So what we achieved is we got rid of that. Remember, there was this summation here over K, which was all the neurons in the system. And that sum is not here anymore. And that sum was our headache, right? Because that made the learning rule non-local. So now the learning rule for feed-forward connections is completely local and therefore biologically plausible.
Mitya,
yeah.
Can
Please.
I say, so here, the price you pay for this is that you have to update this W, right? So that the strength of these lateral synapses must change over time. Is that correct?
But this was also the case in the reconstruction approach. Remember, we said that the lateral connections are given for the Gramian of W. W has to be updated and then put into the Gramian. So that was always the case. When people used, like Olshausen and Field in their algorithm, they used this, and they also used the same kind of lateral connections that were being learned.
So can you say intuitively what is it in the structure of the problem that gives local rules?
It's that all these terms are of this nature where they are kind of like, I shouldn't say they are quartic, but they have four factors in them, and then two of the factors have the same time index, and that allows us to group them and sum over that time index. That's the trick.
Okay.
Yeah, so I should also say that for lateral connections, we also get a local learning rule because WYY has the same kind of sum in the denominator, except this, WYY IJ has YI YJ in there with, indices. Okay, my pen stopped writing. Anyway, so you have the same sum at the top, and so you get a very similar recurrent relation, which is the learning rule that is local in terms of those variables y. So we got rid of all the problems with non-local learning rules that we had before by just redefining the objective function, even though the solution of the objective function is the same. So that's the trick. Any questions? Also, I should say maybe I don't know, Rava, maybe it will help. But for physicists, okay, this is the kind of transformation that is related to the Hubbard-Stratonovich transformation in physics. It's Hubbard-Stratonovich is a little bit more involved, but this is the basic idea. Okay, so that's the trick basically.
So I think I see the mathematical trick, but then this is inherited from the optimization problem, right? So so I'm
Yeah.
trying to think about the significance of the optimization problem now, going back, right? Working backwards.
Right. Well, I mean, to be completely honest,
Mm.
that's how I came up with this objective function. I worked backwards from the learning
Yeah, I was gonna
to
say this is
yeah. No, so what happened was that I got to the cross term, and I said, well, what else can I add to the objective function to make it nice? Because it has to
Uh-huh.
be uh,
Mhm, mhm,
positive, no negative, and so on. And so I added the quartic term, and well, and then okay, it became clear that it should be a
oh
square.
yeah,
So
yeah. It's
anyway, so that's how I work backwards. But I think that now that we have this understanding, this becomes just like a tool in our toolbox, and we've been applying this kind of trick left and right with various constraints in various mathematical changes, and we can get different circuits that implement different computational tasks. So what I showed you so far is just a very simple task, right? The linear dimensionality reduction, which is the top principal subspace or PCA up to rotation, which is implemented by a very basic circuit with linear neurons. But what we can do now is many other computational tasks using the same trick with slightly different circuits. And what I'm going to do in the rest of the talk is show you a couple of examples of this approach. So it's crucial that this slide is clear. So let me know if you have any questions.
I think we're good.
All right. So yeah, so I said, okay, well, this is too easy, right? Because this is just linear dimensionality reduction. And if I can figure out how to advance my PowerPoint, that keeps happening. Okay. Let's see. Sorry about that. Oh, no. Okay, sorry, I have to restart my PowerPoint. So after all this criticism of, yeah, after all this criticism of Apple, Team Windows also has its problems. Okay. Sorry about this. All right. So what we're going to do next is to add a constraint to our objective function, which seems rather innocuous, but actually has important ramification and will allow us to solve more interesting computational tasks than dimensionality reduction. And all I'm doing here is, in addition to optimizing this objective, requiring that the output is non-negative, that y cannot be less than 0. Why are we doing this? Well, our motivation was simply biology, because if you have spiking neurons, their output is characterized by the firing rate, and the firing rate is the number of spikes per second, which cannot be negative, obviously. If you have a graded potential neuron, its output is actually also can be characterized by the number of synaptic vesicles released per second, which is also a number that cannot be negative. So that's why we introduced this constraint. What we didn't know when we added this constraint is that the objective function will lead to a completely different set of solutions, even though it looks like the one that gave us PCA. But with this constraint, it becomes much more interesting. So let me give you an example. Let us assume that the input data are those activity vectors from the fMRI experiment, or the macaque electrophysiology experiment that I showed you in the beginning. So these are these large activity vectors where each component is activity of different neuron or different voxel. And I'm going to simplify the similarity matrix that we saw there with animate and inanimate blocks to just have this, to show this just two examples in each category. And it's important to emphasize that for this objective function to give interesting non-trivial results, it's not enough to require that y is non-negative, but it also has to be the case that x sometimes has negative components. Because if it didn't, then we can have a trivial solution where y is equal to x. So to avoid it, there should be some kind of examples for which the similarity is negative. Okay, so not like this, but like that, right? Pointing in opposite directions. And these are these red blocks here. Okay, so that's the similarity of activities between the animate and the inanimate objects. We assume they're negative. And the ones between same category are assumed to be positive. So in the similarity matching approach, this is the similarity of the inputs, and it has to be matched by the similarity of the outputs. But how do we do that? Well, and also we will require that the rank of the y similarity is 2, so that we just have two-component activity vectors. You can think of this as the output being represented by just 2 neurons. Okay, so if that's the case, then the best we can do to match the input similarity matrix is to have this uniform square with one similarity value everywhere, and we used 1 for that. Okay, just assuming normalization. The same one for the inanimate objects. But then we have to somehow match the similarity in the off-diagonal blocks. But that one is negative. And similarity of y cannot be negative because y is positive. So the products are always positive or 0. And so the best we can do is to have the similarity in the off-diagonal squares, that is between activity patterns elicited by animate and inanimate objects, will be 0. So this is the best solution we can get. But this kind of similarity matrix corresponds to a very simple activity pattern in those 2 neurons, where one neuron, first of all, neurons are binary, but second, one neuron turns on for animate objects, and the other neuron turns on only for inanimate objects. So these neurons are classifiers, and they perform clustering or classification of the input data based on this higher order cognitive categories, animate and inanimate. And that's the similarity matrix that you get that approximates the similarity matrix in the cortex. So if we could run this algorithm on the data, activity data in the cortex, then we could have a classifier that tells you which activity patterns give you animate and which ones give you inanimate objects. And we can prove that this is the optimal solution under some constraints on the data. So in the offline setting, we know that that's always the output, that what this objective function does for these kinds of data is clustering. But our goal was to derive an online algorithm and a neural network that does this in the online setting, and that's what we should do next. And we can follow exactly the same steps that we use to derive PCA from the similarity matching approach. And we use exactly the same trick. And the only difference comes from this non-negativity constraint, where this iteration step involves an additional operation, which is this maximization between the output and the zero, which is just rectification. Okay, so if the value is above 0 or 0, it stays the same. If it's below 0, it's negative, then we set it to 0. People also say that you project the result onto the non-negative quadrant. Okay, that's the only difference. And otherwise, we have exactly the same circuit where we also perform weighted summation of the inputs with the synaptic weights. But after that summation, we put the signal through rectification. This is what they call in machine learning as rectifying linear units, ReLU. And that is not a bad approximation to a biological neuron, which of course rectifies its inputs. And so this is the only change that we need to do to add a nonlinearity that would perform clustering rather than PCA. And it's needless to say, of course, that the learning rules are exactly the same, so they remain local and therefore biologically plausible. Any questions?
It might be stupid, but you said that okay, we put the constraint on y to be positive or 0 and non-negative, so I understand the mathematical problem that is posed if the similarity is always non-negative, but there is also the fact that this makes it unrealistic because again, X was based on firing rate and it has to be positive. So the similarity also, I mean, the actually production between them is also positive. So what I really don't get it. What happens that we still say that it's realistic and biologically plausible?
Very good, very good. This is not a stupid question at all. You're following very well, and you notice something that I glossed over, which is if the learning rates are, if the firing rates are positive, non-negative, then those similarities cannot be negative here. Okay, so the reason we can still do that is because of a little detail that I glossed over. I was going to mention it later to not complicate things. But what we do, if the similarity is still positive, we add a threshold to the objective function. Okay, so that shifts the similarity towards negative values.
Yeah, that's fine. Thank you.
Okay, so thank you very much for that question. You're keeping me honest. Okay, so that's exactly what needs to be done here. Any other questions? Okay, so here is an example of how this kind of similarity-based network is going to do its job. What I'm drawing here is a slightly different kind of network where we added one more step of biological realism, which is the fact that those lateral connections are unlikely to be direct. Because there is so-called Peter's principle, where each neuron expresses only one fast neurotransmitter, and therefore, at least in vertebrates, this is true, can make all excitatory or all inhibitory synapses, but not both, because the long-range projections are usually excitatory. The lateral connections would have to be excitatory too, but we need them to be inhibitory. And so what happens is that we derive a circuit where this lateral inhibition is mediated by this interneuron that is inhibitory. In fact, we didn't even put it in by hand. This came out of the objective function, as you will see in a few minutes, and its activity represents a Lagrange multiplier that enforces a clustering constraint. Okay, so if you know what that is, then that's what it is. If you don't know what Lagrange multipliers are, then don't worry about it, okay? It's just very similar circuit to what we had before. But what I'm doing here is I'm throwing more traditional kind of data onto this circuit, and this is a two-dimensional data set with the data generated by mixture of Gaussians, where the green points are generated by one centroid with the Gaussian noise, and blue by another, and red yet another. And so these three clouds need to be separated or clustered by the circuit. I should also say that these points are presented in an order. It doesn't matter which order to present, and we presented it's the worst for us order, which is like first all blue points as shown here by this digits, and then all green, and then all red. And what I can say happened is that the network clustered this data, and the clustering is indicated by the output activity, where each output neuron represents one of the three clusters. It doesn't matter which represents which, there's spontaneous symmetry breaking. But one neuron responds only to the blue points, the other one to green, and the last one to red. Of course, the network doesn't see the color. It's just there for our convenience so that we can talk about the data. But what's important is that the network, even though it has not seen any data, we just built this network and we just made one pass on the data, and it already classified the data into the three clusters. The classification is categorical or hard in each of those cases, where only one neuron has non-zero activity and there are others zero. So each example is attributed to one cluster, except for the point number 13, where two neurons are active. And it's not hard to see why, because this is point number 13. Because of the noise, it happened to be located halfway between the blue and the red centroids. And therefore, it's represented in a share way by those two cluster centers, because the network couldn't decide which one it should be attributed to. And that's so-called soft clustering, which is a good idea in the intermediate stages of processing before you have to make a final decision, because this way you preserve as much information as possible. So what? Yes.
Here you had to know that you wanted three clusters, right? If you had had a clear dimension, then you would get something different. You would get overlaps between clusters.
Yeah. So that's a good question. Do we need to know the number of clusters beforehand? So there is one parameter in this kind of network, which is the threshold. And that threshold determines the number of clusters. And so we tuned here, there's this one adjustable parameter that was tuned to obtain 3 clusters. In general, you don't have to know the number of clusters. You can build a network with many neurons, and then they will start being recruited into the circuit whenever the new stimulus cannot be represented by the existing cluster centroid. Okay, when it differs from that cluster centroid more than the threshold, like the threshold that someone asked about. Okay, so once that threshold is exceeded, the new neuron is recruited to represent that cluster, and in this way, the number of clusters can be adapted to the data set.
But sorry, but for that you presumably you have to add another constraint, no, to your optimization, because otherwise, what would prevent? Why would the solution not be finer clusters and subdividing one of your clusters into finer clusters?
Yeah, it's one of the determined objective function, like the one that I showed in response to the question about the non-negative similarity.
But effectively, you have some sparsity constraint then on
you can think of this as a sparsity constraint. It's not, yeah, it's not exactly sparse, but you can think of it as a sparsity constraint. It's very similar. Okay, so
I have a question
yes,
regarding
please.
this thresholds. So in your case, you're setting a threshold to kind of get this. How would the network sort of learn the threshold, right? Like how does it determine? How is the threshold determined?
That's a good question. So I don't know. So this is a meta parameter, right? We have to set it by hand. I don't know how it will be done biologically. Maybe that's something that is encoded in the genes. I don't know. It's a good question. Okay, so yeah, so not only does this network do clustering in an online setting, but we can actually also understand how this happens, right? Because each time we present a data point, right, it's a vector in this two-dimensional space, but it's also the set of activities in the upstream neurons, and that vector is projected onto the 3 centroids represented by the 3 neurons, right? And those centroids are encoded in the corresponding synaptic weight vectors of each of those neurons. So this projection is being computed on the presentation of each data point. And then there is this competition through those lateral interconnections where the neurons suppress each other's activity, and the one who has the highest projection actually wins. So that's the cluster that grabs the data point that we see, and it suppresses the activity of all the other cluster centroid represented neurons to 0, except for the case when, you know, there is equal projection, like for point 13, and then they end up with the tie. But this is just the first phase of the algorithm. Remember, there is a second phase where we need to adjust, okay? We need to adjust those centroid based on the presented data points. But you have to adjust typically only one synaptic weight vector, because only one neuron is active in hard clustering case, okay? So that's the only difference from what we had before. But we can actually decompose, you know, how that network does its computation. Any questions?
Well, I was going to ask if you needed a break.
You know, let's see what time it's. I'm happy to do the break, but maybe what I can do, I have prepared some auxiliary slides, and they would be a good slides to show to respond to Stella's question about the nuisance variables. So what we can do, we can declare a break during which I'll show those slides, and those who want, they can stay and listen or watch, but this will not be connected to the rest of the talk. So if you miss it, it's okay too.
Sounds good.
Sounds good?
Yeah, sounds good.
All right.
I sort of want to see the slides.
Okay, so before I do that, actually, I need to show one more slide to set it up, which is what happens when you throw on this network, the same network, you throw data that is not easily clusterable, right? There's not just clouds of points, like natural images, for example. And what happens if you present a series of natural images to a network like this, each neuron actually learns the receptive fields that are similar to neurons in V1, which are these Gabor patches of different orientation and spatial placement. So this is not the first network, of course, that does that. As you probably know, Olshausen and Field in '96 came out with sparse coding network that learns a dictionary like this. Then Bell and Sinovsky came up with ICA that did that. And there were many other papers that produced this Gabor patch receptive fields. But what is important here is this is the first single layer network that does this with local learning rules. So that is biologically plausible learning rules. And that's why this is a significant result, I think. Okay, so now let's do a sort of 5-minute break during which I'll show the slides that will answer Stella's questions. Okay?
Sure.
All right. So the question was, in classifying objects, it's important to be able to ignore some kind of nuisance variables. So for example, in Stella's example, these are sounds that could be spoken by a different person. So the exact frequency composition is different, but the meaning is the same, right? And I'll use an example from the visual cortex, which is face recognition. So the problem of face recognition is difficult because in the pixel level representation, and there was also Rava's question about that, the two faces, the faces of two different individuals could be very similar, more similar than the face of the same individual shown from different angle, right? So two different people shown from the same angle could look more similar in the pixel representation than the same person's face from different angles. And this problem is illustrated here by this manifolds in the high dimensional space, which is the space of pixel intensities, where the idea is that we take all the images of one person's face at different angles and illuminations, and that's the blue manifold. Okay, you can see that it's the same person's face but at different angles. And the red manifold are all the pixel intensity vectors that correspond to another person's face at different orientations. So these two manifolds in the pixel space are highly entangled, meaning that they cannot be separated by a linear classifier. In this drawing, it looks that those manifolds intersect, but that, of course, is not the case. For each image, you can actually say whether this is image of person's A face or person's B face. So they cannot intersect. The reason you see intersections is because what's shown by necessity is a two-dimensional projection of a 100,000 dimensional space. But if you were able to go into that 100,000 dimensional space and explore it, these two manifolds will not intersect, but they will be entangled like this, okay? And that's why you cannot use a linear classifier to solve the face classification problem in the pixel representation. And that's the problem. So one view about how this problem is solved by our visual system is that in the deep architecture, in each layer, the manifolds are being pulled apart without breaking their internal structure, by preserving their neighborhood relationships. But once they're pulled apart far enough, you can just classify them by a linear boundary. And this can be done by many different networks, including our clustering network or anything like that. But if we applied our clustering network straight in the pixel space here, it wouldn't give you anything reasonable. So the question is it possible to sequentially apply similarity matching networks of the kind that I showed and achieve this separation of manifold to classify them linear? And what we were able to show in a toy example, that this is indeed a possibility. So let me demonstrate this example. This is, instead of face manifolds, we just use two spirals that are entangled with each other. And we throw them in this network. And now I have to tell you that this network is the one that has the threshold. Remember that the questions were about? So here is the threshold here. Oops, sorry. So alpha is the threshold. Moreover, we had to modify this objective function to operate not on the inner products, but on this Euclidean distances, because that becomes important when things are entangled. That's what you really care about, the distances, not the angles. Anyways, so we modified this network a little bit, but when we threw this data in the network, it didn't work, meaning that the two neurons grabbed these receptive fields that did represent two opposite spirals, to the two spirals, but did not capture the whole spirals. And if we changed alpha so that those neurons will represent more, then they would include the other spiral. And this didn't work. But it turned out that if we took the same network, but not with 2 neurons, but with 30 neurons, then something very interesting happened. This, just like before, each neuron learned this little receptive field, but now each neuron represented only one spiral, only a fragment of one spiral. But because we had 15 of them, they tiled the whole spiral with overlap, so they even had neighborhood relationships preserved. And no neuron responded to both spirals at once. So they were split into the two groups, one representing one spiral, the other representing the other spiral. This provides a substrate for potentially pulling the spirals apart, and indeed we were able to do that by stacking the same network with the number of neurons reduced at every stage from 64 to 32 to 16, 8, 4, 2. Okay? And below you can see what happens with the similarity matrices, and I'm also showing the spectral embedding of the spirals that are even more entangled now, but they are indeed being pulled apart by this process, so that in the end we can classify them by the linear classifier by this cluster. So we did in fact prove that stacking similarity matching networks allows one to pull apart the manifolds without breaking their structure, just like is hypothesized in the visual cortex. Do I think that this deep learning structure corresponds to the deep learning structure in the visual cortex? I actually don't. And the reason is that it's kind of almost an artifact of our learning process, partially that it's unsupervised, because if we allow supervision, we could do this in just two layers, because once we had half of the neurons represented in one spiral and half the other, then in the next layer we could train the 2-neuron classifier like a perceptron to just separate them. So the reason the network is deep is because of our unsupervised algorithm, and also because we didn't invest any effort into learning those nuisance variables directly, which is what we are working on now. And so that's what I think is necessary to reproduce the human visual system. But in any case, this is an answer of how similarity matching could be used to process the manifolds. So that's all I have for this side topic.
Thanks, Miguel. That's very neat. It's possible, I think, that the kind of things that you show here likely to happen in the initial stage, right? So if you think about the developmental stage, I mean, it essentially is like, as you said, what's missing is that you have to store the representation of the outcome of each steps, and once you do that, then you can. Then the next round you can actually use that outcome. So it's still possible that in the beginning you get like more layers and then you reduce it because you store like each of these things.
Yeah, it could be part of the solution, but by now we are convinced that it's not the whole solution. And so we put a lot of effort into learning these transformations.
Yeah.
And we already have something for learning translations. But I think we should be able to learn many nuisance transformation that you need to identify, say, the same face. You need to learn rotations, even with occlusions and so on.
Mm-hmm.
So that's kind of what we're working on.
Mm-hmm. Thanks.
So Louis, should we?
Yeah, I think as long as you feel up to it, we're good. So
yeah. So let's see. So it's okay. So we have 45 minutes. So that's good. That's good. I think I will even finish early. So if you have any questions, please go ahead and ask. Okay, so let's go. Yes, please.
Maybe one thing that confused me a bit in your spiral example here is that in the 1, 2, 3, 4, 5th stage, why is there this overlap between the spirals?
Oh, okay, very good. Thanks, Rava. So, what this similarity matrix exists in this high dimensional space. Initially 64 dimension and so on. Yeah. So what is shown here is a projection of the data on the top eigenvectors.
I see.
Okay. And it so happens that actually you can see, if you look very carefully in the second, like if you look in here, you can see that there is the very sharp corners here. So the data similarity is already separated in first layer, as we saw in the previous slide. But that separation is not present in the eigenvector dimensions corresponding to high eigenvalues. So if you do spectral embedding in the top 2 dimensions, you're not going to get it. It will be like in dimension 50 something. So it's a very small kind of thing. So what the rest of the network is doing is pulling out that low dimension, that lower eigenvalue. And what I'm plotting every time here is the projection on the top 2 eigenvectors. So the reason that you don't see the separation is because it's in the dimension that is not shown here, but it's already there. And then the reason you have overlap here is because this is 8-dimensional space.
Right, you project. Yeah, now I got it. But it's not in, when you say it's in the 50th dimension, it's not in one dimension. It's presumably in a combination of many high.
Yeah. I actually don't know what the. So that would be what, if we allowed supervision in the next layer, right? Instead of that whole network, we could just like have the first layer that does the first kind of step of clustering with 64 neurons, and the next layer would be just the 2 neurons with the perceptron learning rule, for example. Okay, then I would use supervision to find this direction.
Right. I understand.
And what I'm saying is that direction is orthogonal to the top eigenvectors here.
Yeah. I understand
It
very well.
lives in
Yeah.
this low eigenvalue, subspace.
Mhm. Now, one question. So just intuitively, so the reason why this works is because at each stage, basically, you want this similarity for every pairs of points. So you kind of keep, on the blue strip, you have very close points, and you kind of all of these pairs have to be very similar. That's why you separate the two.
Exactly, that's exactly right, because the network is similarity preserving. So it's trying to preserve neighbor relationships. And the threshold is chosen so that neighbors end up to be only the points that are close enough to be neighbors on the same spiral, not between spirals.
Right.
And
Now, one question is that here you kind of, again, I mean, it's the same question as before for clustering. You kind of knew that you had two objects, and so you went down to 2 dimensions. So what if you
yes,
don't know how many objects you have?
no, that then I don't know what to do. So this parameter alpha, okay, had to be fine-tuned for this to work.
Okay.
Okay, you're absolutely right. And I don't know how to do this in biology.
Okay, thank you.
Okay, so let's go on. If there are no more questions, let's go on to the last topic, which is the two compartment neurons. So please forget everything I told you about the non-negative neurons. We go back to linear problems without the non-negativity constraint. Okay, but we will focus on something rather interesting. I actually don't know if anyone mentioned in this talk, but the pyramidal neurons, which is the most common type of neurons in the cortex, look like this, which is like a tree with branches and leaves and the root system. And those dendrites are called basal and apical. Moreover, these two kinds of dendrites receive inputs from two different sources. So the basal dendrite receive feed-forward input that comes from your sensory organs, from observations. The apical dendrites receive inputs from downstream areas that could correspond to feedback, modulatory instruction input, or prediction, or something like that. And there has been experiments that demonstrate that this is indeed the case. Those dendritic arbors sum those inputs, right, the same weighted summation before, separately in the two compartments, in the basal and apical compartment. And the way those compartments communicate is through the so-called calcium plateaus, which are active processes that are similar to the sodium action potentials, but are mediated by calcium. So if you remember Corey Bargman's talk, she showed recordings from C. elegans neurons, and she mentioned that C. elegans doesn't have sodium potentials, ion channels, and sodium voltage activated ion channels, but it does have calcium voltage activated ion channels. And so those spikes that were recorded in C. elegans neurons were caused by the calcium currents. And this is very similar phenomenon here. And the way this works, if the apical input exceeds a certain threshold, it triggers a calcium signal, just like in C. elegans, that travels towards the cell body. And that's how the two parts communicate with each other. And this is based on the experimental work of Jackie Schiller, Matthew Larkham, Nelson Spruston, and Jeff McGee. And the other very interesting phenomenon that has been discovered is that the plasticity of the synapses, say in the basal dendrite, in this case is non-Hebbian, meaning that the synapses can change their potency without the regular action potentials, sodium action potentials produced by the neuron, because they were able to block those action potentials and they still saw plasticity. And they figured out that that plasticity is due to correlation, just like in the Hebbian case. But it is between the presynaptic activity and the calcium plateau, not the output of the neuron. So that's why they call this learning rule non-Hebbian, but still local learning rule. So this is the more realistic picture of a pyramidal neuron. And it contrasts with the simple picture that we use to derive an Oya neuron, which presumably does PCA. And so the question arises, if this two-compartment neuron is different, what kind of computational algorithm can it perform? And the possibility that we have explored here is that that kind of algorithm is the canonical correlation analysis. It is this algorithm is not as well known as PCA, so I will spend a couple of slides explaining what canonical correlation analysis is. If you know it already, I hope you can bear with me so that I can tell you how this neuron can implement CCA. Any questions so far?
I think we're ready to go.
Okay. All right. So for canonical correlation analysis, we're going to consider an example data set, which is two-dimensional as before, so that we can draw it. And the data appears in the following temporal order. Imagine that there is a clock face, okay? And then there is a hand on that clock face that turns, and the position of that clock face determines the order of data points. Okay, so they appear in sequence like this, all right? And so that you can remember what the order was, I put them all down, indexed by the time step in which those data points were presented. So this is data set 1, but in PCA, that would be it. But in canonical correlation analysis, we deal with data sets, multiple data sets that are synchronized. So here will be the second data set, which will be just like the first one. And the data points now appear in pairs in both data sets at the same time. Okay, like this. And the question that canonical correlation analysis solves is, can you project that data onto a low-dimensional space to maximize correlation? So if you were to project the first data set on the vertical,
I'm not hearing you anymore, Mitya, but I don't know if it's me or...
You know, I am also not hearing Mitya anymore.
Yeah, I think he realizes.
We're still not hearing you somehow.
Mitya, we can't hear you.
But I think he cannot hear us either, it seems.
Mitya.
Of course, he's probably just looking at the screen, us, right? Let's send the chat.
Can somebody annotate a question mark on the screen? Then he'll see.
Yeah,
sorry,
someone send
my sound was off because of
Okay.
electronic interference. And so I could not hear you and you could not hear me. Can you hear me now?
Yes.
Yes.
Okay, sorry about that. Okay, so where did I lose it? Did you hear about the projection onto the vertical line?
We lost you up right there. Yeah.
Okay, so what you can do, you can project that whole two-dimensional data set on one dimension, which is the vertical axis, and then you would have this cosine dependence on time points. And you can project into the same direction the second data set, and that is, of course, achieves optimal correlations of the two data sets, right? So if you align them by time, then they will coincide, and that will give you the highest correlation. Well, here the problem was so tractable that I could like show you the whole thing. So it was obvious that you should project both data sets onto the same direction. But if you did not see the data set like this, which is usually the case, and also if it's online, you might have made a mistake and projected one data set on the vertical line and another one on some other angle. And then those two lines, projections would be out of line, and it will produce suboptimal correlation. So CCA finds those projections WX and WY that achieve maximum correlation in the projected space. Why would anyone need something like this? Well, imagine the following problem. Imagine watching a movie, okay, and your brain has to assign the parts of the soundtrack that correspond to one actor to that actor, and the parts of the soundtrack that correspond to another actor to another actor, okay? Your brain gets two streams of data that are synced with each other, the visual stream and the auditory stream. Okay? But both of those streams are very high dimension. They don't have to have the same number of dimensions, but they're very high dimensional. And all you need to do is to project this high dimensional inputs onto dimensions corresponding to the first actor. Both visual input gets projected to that dimension and auditory input gets projected to that dimension. But you have to find that projection, right? And the way you can find is to adjust the W, the projection matrices, so that the projections are most correlated in time. So that when one actor moves his or her lips, the sound appears. So that's the problem that can be solved by CCA. If there is just one actor, then this is one dimensional projection like is shown here. If there is multiple actors, then this is a multi-dimensional projection onto multi-dimensional space, which is a multi-component canonical correlation analysis. So that's the kind of problem that the brain has to solve, and CCA would be an algorithm to solve it. Any questions? So below, I wrote down a traditional sort of textbook cost function for the CCA, where what you do, you compute the first projection, the second projection, and you compute the covariance that maximizes that needs to be maximized by adjusting WX and WY. Of course, if W's are unconstrained, you can jack up this objective function by just making W's big. To prevent that, there are these two constraints which require that the norm of the projection is 1. That is, the energy in each of the projection is normalized. And this is how CCA is usually defined in textbooks. So what we are going to do is to come up with the neurally plausible algorithm that finds this optimal projections W's in an architecture that is reminiscent of cortical pyramidal cells. And the key to understand how this works is this very simple observation. As I said before, suppose that WY is suboptimal and is pointing sideways. Then the projection onto WY will be displaced from the projection of WX, and the gap will open between the two, which is this mismatch. That means that the correlation will be suboptimal. And so one may guess that the kind of W's that optimize CCA are the ones that close this gap and reduce the mismatch to 0. In fact, this intuition is borne out by exact algebra, because if you write down the mismatch squared objective function and expand it, you find out that these two terms correspond to the constraints, to this fixing the energy of the projection. And this is 1, and this is also 1. So they don't contribute to the optimization, and the only term that matters here is the cross term, which is exactly the textbook definition of CCA. But now that we have this difference squared, just like in the reconstruction approach and in the similarity matching approach, when we had the discrepancy squared, we could derive learning rules that would naturally be implemented by a neural circuit. To see what kind of learning step we will have here, let's just take a derivative of this objective function with respect to wy, and then what happens is that you get this mismatch times y transpose. Okay? So this is the learning rule that we would like to have to learn CCA projections in a neural circuit. Can we implement something like this with our pyramidal neuron? Well, these projections are easy to compute, right? These are just basal or apical inputs weighted by the synapses in the corresponding apical or basal dendritic orbitals. So let's look at that again. So that's the learning rule, and this is the neuron that is going to implement this. So the data set X is supplied to the apical dendrites, and the data set Y is supplied to the basal dendrites. And then the corresponding synapses, through their weights, would project the two data sets on the two currents, the apical current A and the basal current B. These are the two projections. So that's kind of natural to suggest, knowing that these are two compartment neurons and they receive inputs from the two sources that want to be correlated in the projection. Okay? And then what happens next? Well, the apical current is carried by the calcium signal to the cell body, and it added up with the basal current, and they both produce spikes. So this neuron can naturally implement the sum of the two projections and the output that sum, which is fine, but that's not what the learning rule requires. In the learning rule, we have a minus between those two projections because we need to adjust the synaptic weights to minimize the mismatch so that Wx is as close as possible to Wy. How can we switch the sign of one of the inputs? And the resolution to this is shown by this diagram on the right that illustrates the presence of self-inhibition, where the output of a neuron is sent back in an OTAPS. Of course, this is probably not very biologically realistic, so eventually we will put in an inhibitory interneuron that will do this indirectly, okay? Just like we did before to make this biologically realistic. But let's just understand what this kind of feedback does, okay? So what we need to have is the difference of apical minus basal currents. And we are going to use this feedback, Z, that can be computed assuming that the system converged to a steady state, so that the output Z is the sum of the apical current, basal current, and minus this feedback current Z. By solving this with respect to Z, we find that Z is A plus B over 2, which is what we expected, right? That's the sum of the two inputs, and the feedback doesn't just affects the coefficient here. But because we feed back A plus B over 2, then the signal that is coming from the apical dendrite, A, minus the subtracted Z. So this would be, if I multiplied both parts by 2, sorry, I rewrite A minus B as 2A minus A minus B. Okay, this is just like an equivalent rewriting here by adding and subtracting A. but a plus b is the same as 2z. So I can write this learning rule in terms of a minus z. Okay, so this is the trick that allows me to compute this a minus b term without actually having the two input currents of opposite sign, but by passing this feedback into the neuron, and then subtracting z from a, and then computing a minus z, and then the synapses in the basal dendrite have the learning rule which correlates the calcium plateau activity, which is a minus z, and the presynaptic spike, which is y transpose. So this is how we can implement this learning rule using local non-Hebbian plasticity, just like the one that was discovered experimentally. Questions?
So what do you have against using inhibition?
Oh, this is inhibition. This.
I mean, just another inhibitory neuron, right?
You to put the fit for
Yeah, that would,
that could,
right.
So maybe this is also possible. We just wanted to keep kind of in line with the biological data, which says that all those synapses are excitatory, both the apical and the basal. And also the big problem that we have not yet talked about is, it's kind of easy to compute the CCA in a single neuron. And this is not the only way to do this. In fact, we actually could have taken the textbook cost function and also worked with that one. And the neuron would also compute the top CCA component. There are many ways to do one. And in fact, this paper has proposed a similar algorithm, although mapped onto biology differently. And we have also tried to do this with single neurons. The big problem is how to compute multi-component CCA. When you have multiple actors on the screen and you have to compute projections onto each of those dimensions. That's a very, very difficult problem. And we only know how to solve it using a circuit like this one. And that's why I wanted to introduce it. But you're absolutely correct that for a single neuron to compute top component of CCA, you can do it in other ways. Any questions?
I have a question in terms of because this feedback should be some sort of from a previous step, right? If you're thinking of in a more biological sense, then the input wouldn't time with this feedback, right?
So you're absolutely right. This is a very good question. So this is a common feature of pretty much all the algorithms that we talked about. Then when you have multiple channels, the recurrence have to settle before the next data is presented to the network. That is indeed a requirement. And I actually don't know how stringent it is in terms of biology, because some experimentalists, they say that actually it's okay for the previous sort of examples to circulate in the circuit, and others think no, it has to be all over so that there is no overlap. So we actually spent quite a bit of effort to develop algorithms which do not require recurrency, and we have such algorithms for some networks. It's just harder to explain how they work, but this is possible in several cases. Thanks for that question. Okay, so as I just said, the real difficult thing, just like in the reconstruction approach, where Oya proposed how to do PCA in 82, and we came up with the similarity matching network in 2015. Took a lot of effort to solve the multi-channel CCA. Well, maybe not quite a 20-year, whatever, 30-year delay, but still a delay of a couple of years. And but we were able to do this, and this is the sort of how the CCA problem for multiple components is defined. If you open a textbook, don't worry if this is overwhelming, because this is not really crucial, but we use this capital letters, capital boldface letters, to indicate the data matrices where data vectors are concatenated, and also the weight matrices where the weight vectors are concatenated. And then you can write down as maximization of this trace of the projections, and now the constraints, because these are all matrices, become whitening conditions, right? Not just that the energy in each mode is 1, in each projection is 1, but also the off-diagonal terms have to be set to 0. So that's what you do to define multi-channel CCA. But if you try to derive a multi-neuronal algorithm from it, you can't. At least we couldn't find a way. Just like in the reconstruction approach, if you use the reconstruction objective to derive multi-channel circuit, we couldn't, and neither could we here, because the learning rules would come out non-local and so on. So we did the same thing we did before. We went back to the basics and came up with a different objective function, which is heavily based on the similarity terms, like this, and this is the widened similarity, and so on. And so this objective function, I probably shouldn't be explaining where each term came from, so it will take too much time. It doesn't have that much transparency. Suffice it to say that this objective function includes this constraint on the spectral norm of Z, okay? And that constraint is implemented by the Lagrange multiplier n transpose n, where we had to introduce this additional variable that when we solve this optimization problem in the online setting, we came up with a circuit just like before, and this Lagrange multiplier n corresponded to the activity of the interneurons. This is the something I alluded to before. Remember, in the clustering problem, we put in an interneuron, and I said, well, you can derive it from the Lagrange multiplier. This is actually the same trick, and this is one of the ways of systematically derive that inhibitory interneuron. So this is the full circuit in all its glory, and the dynamics is and the learning rules are on the right, and these are the kind of learning rules that we would like to have, that I discussed in the example of a single neuron, except that written for the multineuronal circuit. The WX and WY learning rules are non-Hebbian but local, meaning that the synaptic weights are functions of the presynaptic activity and the calcium plateau, not the output spike rate of a neuron, but the calcium plateau, the internal neuronal communication. And then Q are the synaptic weights between the pyramidal neurons and the interneurons. They're actually just Hebbian. And one thing I should say is that, you know, the Q that we use for the reciprocal connections from pyramidal neurons and back to the to interneurons and back to pyramidal neurons are transposed of each other, but that doesn't mean that then we need them to share weights. Those weights will arise automatically by just following this learning rules, even if you start with somewhat different initial conditions, because there is a convergence of those learning rules. So this will all this all works out, and we confirmed this by simulations. And of course, you know, this is an online algorithm or an objective function that in an offline setting has a closed form solution, which is the CCA, which we know and love. And so we don't really need to simulate this network. We can know what the output is for any kind of data set. Okay, so that's the advantage of the normative approach. And finally, I want to show you the result of the simulating this algorithm, that it actually works and is competitive. Our approach is bio-CCA. It is competitive with other approaches on the market, and K is the number of CCA components, like the number of actors on the screen. And the more components there are, the better our algorithm performs relative to the other algorithms that exist out there. So not only is our algorithm biologically plausible, but it also performs in a competitive way compared to other existing algorithms. And you can read about it in more detail in neural computation, where it just came out. Okay, any questions? Okay, I guess I got everyone.
I have a quick question. In what do you get? What advantage do you get using the similarity? I mean, not advantage, meaning like compared to the previous similarity approach with the different type of neurons, do you get something more with the similarity matching approach as with the CCA?
Well, we just can solve a different computational task. So the way we kind of treat similarity approach now is just like a tool, an instrument that allows us to derive biologically plausible circuits. Right? So the traditional CCA function, which is not written in a similarity-based form, did not yield itself to a multineuronal CCA circuit. But once we were able to write the subjective function using similarity-based terms, right? So all these terms like this, right? Z transpose Z, Y transpose Y, are similarity-based terms, and therefore we know how to deal with them, and there is a straightforward way to convert them to a circuit. That's the trick.
Okay.
I think that's the sort of the utility of the similarity-based approach.
Right. Thanks.
So if you were to go back and try to do this with spikes, I'm struggling with trying to visualize how this would work. I mean, because
yeah. So it's a good question, and I don't have an answer. I can say that my former group member Cengiz Pelivan, who is now a professor at Harvard, actually did look at spiking neurons, and so he might have some ideas of how to do that.
Okay.
But I actually haven't thought about spiking in this context too much. We're thinking about spiking from slightly different approach, but I don't have anything interesting to say in response to this. Okay, so let me summarize my talk then, and this will be my summary slide. So it's finally over. And what I would like to emphasize here is that going back to that Feynman quote that we started with, what theory gave us here is a bridge between biology, so these features that are, known anatomically or physiologically, to the computational task that this feature could solve through the mathematics of the objective function and optimization, where individual properties or individual terms or individual constraints in the objective function map onto certain observations in neuroscience that people know and love. And so you can go back and forth. You can see, okay, well, if we introduce the non-negativity constraint, what is going to happen? Well, you'll have rectifying neurons, and they will learn those clustering or local receptive fields. But you can also go the other way. If you see neural rectification, then maybe there is a non-negativity constraint in the objective function, and so on. Okay, so what this allows you is to go back and forth between the computational tasks that the algorithm solves and the physiology, the learning rules, the neuronal dynamics. So these are now not two distinct things, right? But these are now connected or linked by the mathematics of the normative approach. And that's why I think this deserves your attention. So what I showed you are just some of the examples of this mapping between the computational tasks and the biological reality. There are many more algorithms in this family doing many other things and explaining other physiological observations. And so that is this list of papers. And finally, I would like to thank, of course, all the people that participated in this work, that were members of my group or still are. And these are very, very talented and scientists, and I was really privileged to work with them. Thank you very much.
Thank you, Mitya. Take maybe a couple short questions before we have to go to the life and science discussion.
Yeah, I have a quick question about just conceptually. So, what is the ultimate plan that you have? Is it basically to relate what you just said, to relate observable properties with objective functions, or is it to find one objective function that would from which you could derive as many properties as possible? Where is this tending to, basically? That's my question.
Yeah, so that's a good question. I think eventually, the goal is what you said, that to derive one objective function out of which everything falls out. But this goal may be unachievable, because even in physics, right, there are separate objective function frameworks that are not incompatible, but they're not like, it wouldn't be too meaningful to put them all together. Except actually Feynman had this good example. You write down a mismatch for each equation in physics, you write a mismatch, you add them all up. Okay, so that's the cost function of everything. So, yes, you could do that. But that's kind of an abstract goal, but I don't think a practical one. A practical goal is to keep developing this kind of objective function framework, to try to get more and more insight about what the brain does. And I actually particularly liked what you said in my first lecture, which is, this approach is heavily based on the sensory side, and it's all about reconstructing the world or even matching similarities. But what we really want to get to is behavior. How do sensory inputs generate behavior or help the brain to generate behavior? So we are aiming to have objective functions that include behavior as, dynamical variables in those objective functions. And that's our main theoretical effort right now.
Thanks.
If I may, first of all, thanks, Mitya. I've really enjoyed both of your lectures. Yeah. And if I may comment, I found it very striking that that it is a family of similarity matching networks. And that's because in cognition, similarity is really that governing importance, right? So anytime, or all the way back to the 60s, the 70s, the 80s, and even today, like everything that we talk about in cognition rests on the notion of similarity. Categories and concepts, everything is about similarity and how you define and how you think about the similarity. So that's sort of my comment, being from a cognition perspective. And so in that sense, I think there is it's very interesting that there is that. Second, I want to ask you, how should I pose this question? I'm going to pose it as if, do you think there's any hope using your similarity matching networks to understand how the brain computes similarity that is structural or relational similarity rather than featural similarity? So rather than categorizing animate objects with animate object versus inanimate object with inanimate objects, let's just take a language. If we understand the word on, prepositions on, under, the cup on the table and Stella on the chair have no featural similarity, but has relational similarity, and that's what is required for us to understand and use prepositions like the word on. How would you, and in many ways, cognitively and behaviorally, to compute those structural similarity, we actually have to abandon or forget the featural similarity, because they kind of sometimes contradict each other. So, and I know it's probably still far from going there, but how would you see that problem, like of a structural similarity? Is that within the scope of, could possibly be modeled and achieved by the similarity matching network that you're thinking about right now?
This is a very, very good and deep question, and I don't really have anything intelligent to say about it. I mean, as you know, there are speculations in terms of synchrony and things like that. But I have no clue.
All right, thanks.
Take the floor.
Mitya, maybe another question, which is that if, let's say I take another simpler objective function, like I want to be decoding the value of my stimulus, this should, to some extent at least, subsume similarity. Because if I decode perfectly, then I will also respect similarity. So, but then there is something about the nature of the errors that is different when I use one objective function or the other objective function, that kind of changes the solution. And I don't really have a good intuition on what is it really that is different. So maybe you can have some intuition you could talk about.
Well, I actually, it's a good question. As I said, as long as in the linear problem without the non-negativity constraint, the two objective functions have the same solutions. So it's just the energy surface that is different. And why did one work and the other didn't? Algebraically, I can tell why this is. It's because of those, the biquadratic terms, as I would say, where you have x transpose x, y transpose y. So that's what happened, or y transpose y transpose y. So that's how the magic happened. Is there a deeper kind of reason? I don't know. I mean, the most utilitarian view of all this is that we just came up with this formulation that through this, Hubbard-Strudanovic-like transformation allows you to circumvent the non-locality problem. But, there may not be any more depth to it. On the other hand, there is this representational similarity analysis from Krieger-Skordi that I showed, and the work that Stella mentioned from psychology, which is all like based on similarity, and similarity is important. And maybe there is some more, some depth to it, other than just like a mathematical trick. So I don't really know.
So maybe let me ask the question differently. So if you took, say, a stimulus set with like all animals, let's say, and then you constructed a network based on this similarity matching, would you then get cells that would be like informative of whether you have a dog or a cat? So would you get cells that tell you about the identity also of the stimulus just because you ask the similarity to be preserved, or no?
Well, I mean, you're asking how far along can you get using simply unsupervised methods, which is a valid question, and I think you can get pretty far. I don't know if you can get all the way, but you would have to. I think that what is necessary if you're going embarking on this kind of direction, which is kind of what we did, is to include this what they call now self-supervised approach. The fact that similarity isn't just between different objects, but there is a similarity in the visual field, like between different pixels of the same image, or similarity in the temporal stream between sequential frames, right? They're not randomly chosen. There is some continuity to them. So if you take into account that similarity, you can do all this what you described, from what I understand, in based on the work in self-supervised learning.
Okay.
Can I just follow up on Rafa's question? So given the same thing that Rafa said, would your model be able to, would your model prediction would be then it's easier to separate cats from turtles than cats from dogs using the similarity matching network?
I think the answer is yes, but
Okay.
not a single layer one.
That's
Right?
yes.
Not on the pixel level, because
Yes, not on the pixel level.
on the pixel level, two cats and there. I'm sure there is a breed of dog and a cat that look more similar than two different breeds of dogs.
Yes.
So it would have to be a higher order feature, and then the question is, how do you learn those higher order features? And I think only partly this can be answered by similarity. I think you need to do something with this transformation invariance learning, which requires additional ideas. Okay. So yeah, so that is needed. I think that's sorely missing.
I think I saw Eric coming into the room, so we're basically ready for the so. But let's thank Mitya again. I think it's been a great series of talks.
Thank you.
So where's Eric? Trying to find him.
Good morning, I'm here. Yes, good evening.